Hey, good. How you doing? It's this chapter 11. We're doing DNA replication and recombination. Most of this is stuff you already know. So I have it cut into two different segments. One of it is just introducing it and talking about bacterial replication. Then part two is really me talking about how you learned replication in Bio 1 with all of the enzymes, and you really learned bacterial replication. The second part of video 2 is looking at how it differs in eukaryotic replication. I feel like that is where you're going to get new information. So take that with a grain of salt. I would watch this with a beginner's mind. Maybe you'll remember some things you didn't know you forgot. And then on to the last part of the second video, I think will be completely new information for you. So to get started, I'm going to do in video 1 here, we're going to talk about the process of semi-conservative DNA replication, which is the way it works, and I'll explain that. The polymerases and all the enzymes involved in bacterial replication, and I'm going to look at some of the consequences of their lack of function. Assume that if they can't do their job, the thing that they are supposed to do does not happen, and then every consequence downstream of that, the next things that would happen, are also now an issue for that replication process. In part 2, I'll also continue on with some of the consequences of lack of function. So we already talked a little bit about the structure of DNA and how the structure of DNA really does show the way it is copied. It's two matching strands of DNA that run anti-parallel, but they are complementary, meaning that every adenine is complementary. It matches with a thymine. We had pyrimidines and purines that always match together. Adenine and guanine were purines. They were pure as gold. Thymine and cytosine were pyrimidines. They were a little bit smaller molecules than the purines. I've kind of reviewed all of that. And now I also showed you that that structure itself contributes to the accuracy of how DNA actually replicates. So if I'm going to replicate my DNA, I pull it apart. I have now a opening on each strand is now missing its complement. And then I build that complementary sequence using a series of enzymes in an anti-parallel, running the opposite direction, but complementary manner. So a quick review of some of the things we have talked about so far. The information in genes gets passed on into a new generation. So this DNA is actually containing your information, but also the information, the code you're passing on to your offspring. And this is also where variability comes from. So the fact that you and I are different from each other, or Alice the bird who is just tweeting away this morning and I are different, is due because we have different code inside of our DNA, and that causes us to make different proteins. In order to be genetic material, the genetic material must be able to copy itself accurately. It must be able to replicate. That's today's lecture. It needs to be able to store information, so it has to have a code that is readable by some other part of the body. That has to be accessible, meaning we can turn it into proteins or whatever it's supposed to code for. And there has to be a mechanism for that information to vary from individual to individual. That's going to be mutation, where new alleles, new sequences of code that code for specific genes arise. DNA is replicated in a semi-conservative manner. If you want to go back and read about the history of this, there's a section in your textbook, but I've never understood why we do a big segment on a bunch of things that we know are not true. So I'm just going to highlight the fact that semi-conservative is, after some experimentation, is the way that we know that this process works. What that means is that the strand itself, it's this one, the strand separates into two different strands, and then the new DNA, the new nucleotides, are laid down, each with one original strand and one new strand matching it. If it was conservative, I would take my strand apart, copy it, and then make an entire new daughter strand. This would be a source of a lot of error. So we know that that doesn't happen, and we know because we've tagged stuff with radioactive material. If it was dispersive, we would open it up and kind of copy specific genes. You see how these are blue, and the blue ones are representing new things. This would also be a pretty good source of error. It was a big concern when people were trying to understand the process itself, was how could this process possibly work in a manner that minimized errors but sped up the copying process? The only way that that is actually going to function in a way where we keep accuracy is the semi-conservative mechanism. This semi-conservative mechanisms, and I have a little bit in your homework for you to look at this as well. It should be just, I know everybody loves the drop and slide things, but those are my only options in the homework. I want you to look at Mendelssohn and Stahl's experiment because it comes up in other places. People build on this experiment. Mendelssohn Stahl in 1958 took some DNA, this is E. coli DNA, and they labeled it with nitrogen 15. That nitrogen 15 was built into this organism's DNA, so all of the nitrogen in its DNA was nitrogen 15. They then added nitrogen 14, which is the normal nitrogen, to the generations of bacteria and let them replicate. And what they found as they went through the experiment was that it showed semi-conservative replication. Each new daughter piece of DNA was built out of the solution that was available for nitrogen 14. And so each new replicated strand ended up having 1/2 nitrogen 15, 1/2 nitrogen 14. And as the generations passed, and we'll look at this a little bit more as we get into the process of PCR in the lab, as the generations passed, those original pink parent strands, the ones with the 15, nitrogen 15, remained 50-50 as part of strands, but the new DNA was being built from each daughter and copying, so most of the DNA strands were completely devoid of nitrogen 15 and had only nitrogen 14 in it. If this is your thing and you're looking at trying to understand the process of DNA replication in extreme depth, this is actually the paper that they published. It's fairly famous in its simplicity. It's also been demonstrated in eukaryotes. Semi-conservative replication seems to be the standard across the entirety of DNA replication, even for morning spring birds who are, I'm sorry, it is spring today. They are demonstrated in eukaryotes in Vinca faba. This is a type of bean, like a bean that we would eat. Taylor, Woods, and Hughes showed in 1956 using thymine and the same very similar mechanism. They tagged thymidines in the DNA that was about to be replicated, and then they watched the process of meiosis and traced the process through, showing that in each case the parent strands started out together, they were pulled apart, and then here's the next step, and then the new daughter strands are built from new nucleotides matching the original parent strands. And this is their paper. When we talk about DNA replication, there's a couple of words that I want you to have under your belt before we actually start the process. We start DNA replication at the origin of replication. This is the place where replication starts. I'm going to label it ORI. So this is the origin of replication on this bacterial chromosome. This thing is going to be the place where we're actually opening up the DNA to start the process. When I do that, I open up a replication bubble. At the origin of replication, and it forms two forks or replication forks. Here is a fork, and here is a fork. And DNA replication will proceed this way around the molecule and simultaneously this way around the molecule until they kind of copy the entire thing, and I pop two new freshly replicated strands of my bacterial DNA. The bacteria then go through the process of splitting apart. This is binary fission. Bacteria have one origin of replication. It's called the origin of replication C. In eukaryotes, when we look at it at the end of video 2, eukaryotes have multiple origins of replication. One of the things I'll talk about in a little bit is, and something to look for, is because adenine and thymine have only 2 hydrogen bonds, whereas guanine and cytosine have 3 hydrogen bonds. What we find often at places where the molecule of DNA needs to be opened up, we find large sequences of A's and T's together, or A, T, 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 because in this case, I have fewer hydrogen bonds that actually have to be separated away from each other. So these regions where I have lots of A's and T's are easier to open up, easier to be an origin. We're a place where we start transcription later. Replication is bidirectional, meaning it's going off in 2 different directions simultaneously. The entire sequence that gets replicated is called a replicon. In E. coli, the replicon is the entirety of the DNA. In eukaryotes, a replicon is not going to be the entirety of the DNA because we have many different origins and replicate until you run into another origin. So each replicon is much, much shorter. In E. coli, however, it's the entire genome. They replicate all the way around until the 2 new pieces of DNA pop away from each other. Involved in this are a lot of different polymerases. I think in Bio 1, you probably learned about DNA polymerase 3. You may have just called it DNA polymerase or DNA pol. DNA polymerase 3 is the one that plays the biggest role in elongating the new daughter strand. We're going to talk about 1, 2, 3, 4, and 5, but we really are going to highlight 1 and 3 as part of this lecture. I'm going to shorten DNA polymerase to pol here. It's as long as we establish what enzyme we're talking about, this is a completely reasonable way to talk about it. So we'll talk about a lot about DNA pol 1 and DNA pol 2, DNA polymerase 1 and DNA polymerase 2, but I'll highlight some of the others too also. DNA polymerase 1 is an enzyme with very few, it's tiny, it has very few, this is amino acids, and it really directs the DNA synthesis at the start. It does require a template, and we'll talk about what that means, but it requires an established parental strand that it is building and it's reading and then matching its nucleotides to. It requires the presence of all 4 DNTPs. These are adenines, thymines, cytosines, and guanines that are freely floating around in the cytoplasm or the nucleoplasm in our case, and are available for building. They have DNA nucleotide triphosphates. They are a nucleotide with 3 phosphates attached, and some magnesium is required in order to catalyze this process. DNA polymerase 1 will elongate the new daughter strand in a 5-3 direction. This is going to be a rule that we're going to build on. We always build in 5-3, and we always, with DNA polymerases, add to the 3' end of the growing molecule. DNA polymerase 1 is small. It can fit into small areas, and it can build as long as it has a template. DNA polymerase 1 elongates the new daughter strand in a 5-3 direction, always adding to the 3' end. And if you go back and look at your molecules, what this is really talking about from the last lecture is the sugar inside. I'm drawing the DNA sugar right here. So this is a deoxyribose. The deoxyribose sugar here is it's a penta, a 5 carbon sugar, and it has 1, 2, 3, 4, 5 carbons. This is carbon 1, carbon 2, carbon 3, carbon 3, carbon 4, and carbon 5. When we say 5 to 3, we're talking about the directionality of the molecule. We say I'm building on the 3' end. I am saying that in order to add a new nucleotide to an elongating strand, I need to put the new one here on the side with the carbon 3 and this hydroxyl group right here, this OH. If I built the other way from 3 to 5, I would be saying I was building on top of it. I can't really draw it here without running off the screen. I can't build here for a variety of reasons, but functionally because the enzyme itself can't build on that side of the molecule. When I have a growing chain, this is that deoxyribose sugar right here attached at its 1 carbon to its nitrogenous base. It's an adenine right here. 1, 2, 3, 4, and there's a carbon up here, 5. I am building on this side of the molecule every single time. When I want to add a new nucleotide, I need to add it as a nucleoside triphosphate, 1, 2, 3 phosphates on this free floating thing. It's floating in the cytoplasm or cytosol. The enzyme polymerase, and in this case a DNA polymerase, grabs it, it attaches it here, breaks it here, and attaches, I'm going to circle it here, here is the same thing. This attaches this molecule to the free 3 side of the existing growing elongating strand. And then 2 of these phosphates are popped off. This is a pyrophosphate, for those of you who've taken orgo. And there's a little bit of available energy that we can use to catalyze this. That energy that catalyzes this reaction, that bond that forms here is the new, this is a mess. Let me change colors. This one right here, is the new phosphodiester bond. This monomer is attached to the growing elongating strand on the 3' end by adding a nucleoside triphosphate, popping off 2 P's, 2 phosphate groups, and forming a phosphodiester bond. DNA polymerase 1, 2, and 3 elongate an existing strand as long as there is a primer. It cannot initiate elongation on its own. And what that means is there needs to be a molecule of DNA with a free 3 side, or a molecule of RNA with a free 3 side, in order to bind the next nucleotide to. These guys are really limited in the fact that they cannot build without a free 3 side. A primer in DNA replication is going to be laid down by an enzyme that puts down a piece of RNA. When we do it in the lab for PCR, we're going to have primers that come in little tubes. We'll add, those are going to be DNA. So I'm just leaving this as a primer in general. A primer is a thing that primes the surface for attachment. If you've ever painted a house or a room, you lay down a thick coat of paint primer, and that allows the coat that you're actually interested in coloring your house with, the final paint, to stick better. If you're doing a lot of makeup and you have full face makeup, a lot of times you put a primer down to make sure that the makeup sticks and stays, it doesn't slide off. Primers prime the spot for attachment. DNA polymerase 1, 2, and 3 need the spot primed for attachment before they're allowed to attach. All 3 of them can also remove segments of DNA that are sort of bonded inappropriately. Maybe the molecule is structurally a little bit too wiggly. I'll show you how some of that can look. They can excise. This means to cut out incorrect nucleotides. But only DNA polymerase can work on the 5 to 3 region, as it excises primers made out of RNA. and then fills in the gap with DNA. DNA polymerase 1, 2, 4, and 5, so not 3, can initiate repair if the DNA itself is already broken. So we're talking about the strand and the phosphodiester bonds. If they're broken, everybody but 3 can fix it. I want to show you a little bit of what DNA polymerase 3 we envision it looks like as an enzyme in bacteria. It's a holoenzyme, which means it's comprised of multiple enzyme complexes. It's more of a molecular machine made out of pieces of enzymes that work together, subunits, in order to create the entire process of DNA replication. There is an alpha unit here. There is an alpha unit here. This alpha that are doing the actual polymer building. Polymer ization means the action of doing the thing. They're building the DNA polymer new strands. There is epsilon that is involved as an exonuclease. This is removing errors, replacing errors, and then the core assembly that is holding it all together. I am not going to ask you to identify these on an exam or a quiz or a homework. I want you to recognize that this thing is made up of lots of different enzymes working together. It doesn't work as an individual piece. This is a machine, kind of like ATP synthase when you did metabolism in Bio 1. That's a thing that does a really complicated function. It makes ATP. It's made up of lots of enzymes working together. This is made up of lots of enzymes working together. I have a little bit of practice with me. I know that's short. The second part is really long because it doesn't make sense to break partway through the next part. So bear with me as video 2 gets a little bit longer than this. I would like you to ask you questions basically at this point. What is a polymerase and what do they do? So all known bacteria DNA polymerases, 1, 2, 3, 4, and 5, what can they do? And I'll give you a second. This is where your 5 to 3s come in. I always remember, because I am building on the free 3, that kind of rhymes and it's hard to say for me. The free 3 is where we're building, which means all polymerases have at least the ability to build on the 3' side. They have a 5 to 3 polymerization activity. This here, it's I'm going to include it as a link inside of this week's help too. This is the book's little movie about DNA replication. I also have some from Khan Academy that I think you will find helpful. Well, I guess I kind of do want you to get a little bit bogged down in how the machine works. If you watch the videos, and then we can talk about how that machine works to actually polymerize the DNA. So here is this media thing. I made it in here so that you can link if you actually have the slides, and I'll post the Khan Academy one separately in our Canvas site. With that, I will go on to video 2. You can take a break or come back later, or you can just keep watching. I upload them separately because I think it's good for you to take a break. All right, I'll see you in the next video, and I'll see you in class. All right, bye-bye.